of inheritance overview video. Gregor Mendel was able to determine an important principle of heredity, which is that traits are governed by discrete units known as alleles, and that there are 2 alleles per gene, and these alleles segregate equally during gamete formation. However, he had no idea where these alleles, or loci, were actually located, just that they were found in the gametes, the egg and sperm, and that fertilization united them randomly. Some years later, using Drosophila as an experimental model, Thomas Hunt Morgan and his graduate students were able to actually show where alleles, or loci, the locus of a gene, is found on chromosomes, and that genes located closely together on the same chromosome do not assort independently. The fruit fly, Drosophila, is an excellent animal model for genetics because experimental crosses can be readily manipulated. The flies have a short generation time, and they also produce a lot of offspring for statistical purposes. Morgan observed a few rare flies had the distinct phenotype of a white eye. He referred to this as a mutant type, as opposed to the very common type found in nature that he called the wild type, and that was a red eye. So he was interested about this white eye, and he decided to cross pure breeding red eye females by pure breeding white-eyed males. The F1 generation all had red eyes, showing that red is the dominant type to white. However, the F2 generation exhibited a very interesting pattern in that only the females had red eyes, but half of the males had white eyes. This was different than what he had expected. When Morgan performed a reciprocal cross, a white-eyed female by a red-eyed male, the F1 generation showed another interesting pattern of all white-eyed males and all wild-type females. The F2 crosses resulted in half of the females with white eyes and half of the males with white eyes. Further, looking at the Drosophila chromosomes, Morgan noted that males had one chromosome that looked different than the females. He was able to deduce that these are sex chromosomes that he called X and Y, and that during meiosis, males produce gametes with half containing an X chromosome and the other half containing a Y chromosome. The female has 2 X chromosomes. Therefore, all eggs will normally carry a single X chromosome. The fertilized eggs bearing X spermatozoa give females 2 X's, and those fertilized by Y bearing spermatozoa give males an X and a Y. Once Morgan discovered that genes are located on chromosomes, he began to suspect a possible conflict in Mendel's principle of independent assortment. Morgan's students were able to show that 2 or more genes can be located on the X chromosome, and therefore they are physically linked together. Closely linked genes are not free to undergo independent assortment. Instead, alleles at all the loci in the linkage group will be transferred as a single unit during gamete formation. However, if the genes are located far enough apart on the same chromosome, there can be the appearance of independent assortment due to crossing over. This is also known as recombination. The degree of crossing over between any 2 loci on a single chromosome is proportional to the distance between them. That is, the further apart they are, the greater the likelihood that recombination will occur. Humans have 23 pairs of homologous chromosomes, and they can be studied via a karyotype. A karyotype is used to look for abnormal numbers or structures of chromosomes, often during pregnancy. Sampling can be done through either an amniocentesis or chorionic villi sampling. Chromosomal abnormalities can cause problems due to the presence of either too many or the absence of important alleles. One common way they can occur is during meiosis, when there is a failure for the chromosomes to separate properly. This is known as non-disjunction. It can happen during anaphase 1 or anaphase 2 with slightly different results. As a woman ages, the likelihood of non-disjunction occurring increases. Non-disjunction will result in gametes having either an extra chromosome, this is known as a trisomy, or the absence of a chromosome, known as a monosomy. Zygotes with monosomic chromosomes usually result in death. However, trisomies are not as lethal or problematic. A common trisomy seen in humans is trisomy 21, also known as Down syndrome. Let's look a little bit more closely at molecular inheritance. We know now that a gene is a sequence of DNA that specifies one or more RNAs or proteins. Thus, DNA is the genetic material. The search for the genetic material was an intense one, and it wasn't until a series of experiments were conducted and a structural analysis of DNA completed that researchers accepted that DNA was the genetic material. They demonstrated how DNA fulfills the requirements for genetic material. 1, DNA encodes information in the form of the 4 bases. 2, DNA is susceptible to mutation. But 3, DNA can be faithfully and precisely replicated. And 4, the process of DNA replication is one that shows you it can be readily transmitted into daughter cells. DNA replication starts at the origin of the replication site. The origin are specific stretches of DNA. Several proteins are involved in the formation of the replication fork at the origin. This process is known as initiation. The proteins that you need to know are topoisomerase, helicase, single-stranded binding proteins, and primase. The replication fork is unwound, and once that has been unwound, sections of the parental DNA strands are available to serve as templates for the synthesis of new complementary DNA strands. This is known as semi-conservative replication. A primer, via primase, is needed to provide a free end for new bases to be added. This is the 3' OHN. Enzymes called DNA polymerases catalyze the synthesis of new DNA by adding nucleotides to the 3' end of a pre-existing chain. For the leading strand, only 1 primer is needed for DNA polymerase 3 to synthesize the entire strand. This is known as the leading strand. Elongation happens, again, in the 5' to 3' direction. Therefore, DNA polymerase 3 must work along the other template strand in the direction away from the replication fork. The DNA strand elongating in this direction is called the lagging strand, and it results in Okazaki fragments due to discontinuous synthesis. Because of this, primase is needed multiple times, and when the primase is removed, DNA pol 1 must fill in the gap. The enzyme DNA ligase will join the ends of these fragments together. DNA polymerase 3 is not perfect, and errors do happen approximately 1 out of every 100,000 bases. But luckily, those errors can be fixed by an array of DNA repair mechanisms. It's also important to note that there is an end of the chromosome replication problem for eukaryotes. Because eukaryotes have linear chromosomes, the replication machinery can't complete the 5' end of the daughter strand because there is no 3' for a pre-existing polynucleotide to be added to. Over successive replication cycles, the chromosomes then will get shorter. However, eukaryotic chromosomes do have special nucleotide sequences at the ends of chromosomes called telomeres. These telomeres don't contain genes. Instead, they have multiple repetitions of sequences that act as a buffer zone protecting the genes. Our gametes have a special enzyme that helps to catalyze the length of the telomere. This enzyme is known as telomerase. Telomerase is inactive in somatic cells, but in some cancer cells, it has been activated. Finally, when we look at DNA and how it's packaged to fit inside the nucleus and then condense into chromosomes, you can see that it involves a very complex systematic packaging mechanism, not yet fully understood. Double-stranded DNA is first wound around histones, special proteins that interact directly with the DNA. These histones, along with the DNA, form the nucleosome, the basic unit of DNA packaging. The next level of packing involves additional folding to form a 30 nanometer fiber in diameter. However, to further condensation, the 30 nanometer fiber forms loops called loop domains, and then this forms a scaffolding that is folded onto itself and coiled into a more compact chromosome. These are now characteristic of the chromosome seen in the M phase of the cell cycle. Great. We've gone over how DNA molecules are arranged in a chromosome, how DNA replication provides the copies of genes the parents then pass on to the offspring. And again, hopefully you have a better understanding of both how chromosomal and molecular inheritance works. Have a great day.